to Inside Chlorpromazine, the podcast where we unravel the unexpected medical potential of a well-known antipsychotic. If you've been following along, you know we have explored chlorpromazine's effects on immune modulation, inflammation, and even viral entry mechanisms. Today, we are getting even closer to the virus itself. This episode is titled Hijacking the Hijacker, Chlorpromazine Interference with Viral Replication. I'm joined once again by a guest, a microbiology researcher with deep insights into how viruses like SARS-CoV-2 replicate inside host cells. Welcome back, Chythanya.
Thanks, Sowmya. Excited to explore this layer of the chlorpromazine story. Viral replication is where the real battle takes place, and chlorpromazine has some intriguing effects here.
To set the stage, could you briefly walk us through what viral replication typically looks like, particularly in coronaviruses?
Sure. Once a virus enters a host cell, usually through endocytosis or membrane fusion, it starts the replication process. For SARS-CoV-2, the virus releases its RNA genome into the cytoplasm. From there, the host ribosomes translate viral proteins. Then the virus assembles replication complexes. Then new copies of RNA are synthesized. Structural proteins are assembled and the virus reforms. And finally, new virons are released via the Golgi or lysosomal pathway. So each step relies heavily on hijacking the host cell machinery, and this is where drugs like chlorpromazine comes into the picture.
We have talked about chlorpromazine's role in disrupting clathrin-mediated endocytosis. Is there a link between that and replication?
Yes, absolutely there. Chlorpromazine inhibits clathrin-mediated endocytosis, which is one of the pathways many viruses use to enter cells. But this inhibition also indirectly affects replication because certain viral components rely on endosomal transport even after entry. So, by disrupting endosomal trafficking and intracellular membrane organization, chlorpromazine can interfere with how viral proteins are processed, transported, and assembled.
That's interesting. So the drug interrupts the virus' internal logistics.
Exactly. You can think of viral replication as a very efficient assembly line, and chlorpromazine throws a wrench into the machinery. Studies have shown that chlorpromazine interferes with endosome maturation, Golgi processing, and even lysosomal recycling, all of which are critical for forming new virus particles.
Do we know if chlorpromazine has a direct effect on RNA replication?
There is evidence that chlorpromazine can reduce viral RNA levels in infected cells. In cell cultures, researchers have observed decreased SARS-CoV-2 RNA expression when treated with chlorpromazine. This could due to multiple factors. First one, inhibited formation of replication organelles. Other, disrupted lipid membrane integrity which affects replication complexes. Altered cell signaling pathway needed for viral RNA synthesis. But to be clear, the mechanism is fully not understood. It's likely a combination of effects rather than one single thing.
Is chlorpromazine's impact on replication unique to coronaviruses?
Not at all. Chlorpromazine has shown inhibitory effects on the replication of multiple viruses, including influenza, Ebola, dengue, hepatitis C. This suggests that its action targets host-dependent pathways that many viruses exploit. That makes it particularly attractive as a broad-spectrum antiviral, something we desperately needed, especially in pandemics.
Now, I have to ask, if chlorpromazine is interfering with host cell processes, is the risk of toxicity?
There is a valid concern. Chlorpromazine does effective cellular trafficking and that can impact normal cell function. However, the doses used in antiviral studies are generally lower than psychiatric doses, and short-term exposure may carry less risk. In vitro studies shows that antiviral window where the drug is effective without being toxic does exist, but it would need careful dose optimization in clinical settings.
Could disrupting replication pathways be relevant to long COVID, especially if residual viral material plays a role?
That's an existing possibility. Some researchers believe persistent viral reservoirs or remnants could trigger long-term symptoms. If chlorpromazine can limit replication or help clear these remnants, it may have a role in reducing viral persistence. Again, we need more data, but the data is being explored in post-viral syndrome and warrants deeper investigation.
So, in light of everything we have covered, what makes chlorpromazine a compelling candidate for repurposing?
It's already FDA approved. It has broad antiviral potential and it targets host pathways that are less prone to viral mutations. That means it's less likely to become obsolete due to variants, a major problem with spike protein targeted therapies. Plus, it has a multimodal action antiviral, immunomodulatory, and anti inflammatory. In the complex pathophysiology of COVID-19, this kind of polypharmacology is very attractive.
To wrap up, chlorpromazine appears to interfere with viral replication not by targeting the virus directly, but by disrupting the host cell's ability to support viral assembly and RNA synthesis. It's like sabotaging the factory the virus has taken over.
Perfectly put. It's not a silver bullet, but it's fascinating candidate in the world of host-targeted antiviral therapies.
Thank you, Chaitanya, for unpacking this technical but critical topic. In our next episode, we'll explore chlorpromazine's role in autophagy and cellular recycling and how that may impact viral clearance and inflammation. Until then, stay curious, stay informed, and thanks for tuning into Inside Chlorpromazine. Thank you.